Friends, welcome to Biology Made Easy. Today we will discuss mitosis. Mitosis is a type of cell division which is also known as equational cell division because the two daughter cells which are produced, they are equal in all respects. That is why it is otherwise known as equational cell division. Mitosis was first observed in case of plant cells by Strasburger and in case of animal cells by Fleming. But the name mitosis was given by Fleming. Now where this mitosis takes place? Mitosis usually takes place in somatic cells, that means in the all body cells. That is why mitosis is also sometimes known as somatic cell division. But it can take place in reproductive cells only during the growth of reproductive organs, not at the time of production of gametes, sperms, ovum, like that. Next thing, in case of animals, it always takes place in diploid cells, while in case of plants, it takes place both in diploid cells as well as haploid cells, because there are some lower plants like algae and bryophyta and the part of pteridophyta where there are gametophytes. So there the mitosis takes place. So this is the difference between plant and animal cells where it takes place. Now suppose we want to study mitosis in classroom, then which is the best material for it? The best material is onion root tip. Onion root tip. Because in case of onion there are 2n is equal to 16 chromosomes. Very less number of chromosomes. Plant chromosomes are bigger in size. Again, onion is coming under monocot. Monocot chromosomes are bigger than dicots. That is why onion root tip is commonly taken for demonstration of a mitosis to observe chromosomes during mitosis. But if we study animal cells, in case of animal cells, we can take bone marrow cells, and also we can take a nail base. You can see at the base of the nail it is slightly white. So from that portion also we can take cells. So this is how we can study mitosis. Now mitosis can be discussed under two separate headings. In the first step, the nucleus divides. That is known as karyokinesis. Karyokinesis, and the other is cytokinesis. Karyo means karyon means nucleus. That means the nucleus will divide. Cytokinesis means cytoplasm. The cytoplasm will divide. Now the karyokinesis is a continuous process, but for the purpose of our discussion, the karyokinesis we can divide into 4 stages. First is prophase, number 2 metaphase, third one is anaphase, the fourth is telophase. They take place in a sequential manner. Now let us discuss first this prophase stage. Prophase stage is the first stage of mitosis. That is why this is a very long stage, or you can tell it is the longest stage in mitosis. And this is the first stage. This is the first stage and the longest stage of mitosis. Now during this mitosis, what happens? Just before this mitosis, there is the cell cycle, that is G1S and G2. The chromosomes have been already duplicated. The chromosomes are very long, thin and uncoiled. So now what will happen? Each chromosome is still long, but each chromosome has 2 chromatids. And these 2 chromatids, they are intertwined around to each other, forming different types of major and minor coils. So the next point you can write, each chromosome has two chromatids intertwined around each other, forming coils. So suppose this is one cell, and within this cell, this is the nuclear membrane and nucleus, and within this the nucleolus is there. So now the chromosomes which are present, they are like this, not so much distinct like this, but each chromosome has two chromatids attached at the centromere. Now at this stage, the whole nucleus looks like a ball of wool, which is also known as spireme stage. So next point you can write, nucleus looks like a ball of wool, which is known as spireme stage. Now when the division of the chromosomes, the nucleus will start. Now initially the chromosomes are very long. Now due to presence of different types of proteins like condensin, cohesin, there will be contraction and spiralization. So during this process, what happens? First there will be dehydration. That means the nucleus will be dehydrated. As a result, what will happen? The viscosity and refractivity of the cell will increase. Dehydration leading to increase in viscosity and refractivity. Then next point, what is? Now due to presence of this condensin from the name you can guess, there will be condensation. And the protein cohesin means there are now two chromatids, these two will be together, they will be there together, they will not be separated, they will be separated in different stages. So these two proteins, they play a very major role. Now due to this condensin protein, the entire chromosome will condense and there will be spiralization like this. So when there is condensation and spiralization, what will happen? The length of the chromosome will decrease. And to what extent it will decrease? It will decrease to 4% of the entire chromosome size. Condensation and spiralization leading to 4% of chromosome size, or you can tell 1/25 of the size. Suppose the length of the chromosome was 100 micrometer, now it will be 4 micrometer. That means to that extent it will condense due to spiralization. As a result, the chromosomes will be little bit clear, because in the interphase they were not clear. So now they will be, to some extent, they will clear because of the condensation and spiralization. Now side by side, some other things happen. If it is an animal cell, centrioles are present. So suppose here two centrioles are present. These are the centrioles. At this stage, the centrioles will replicate. So next point is, in animal cells, centriole replicates and move to either poles. Now they will move to either poles. Now what is the function of these centrioles? The centrioles is usually, their main function is they will give rise to spindle fibers. So suppose this is one centriole and it will give rise to spindle fibers, and these spindle fibers will be attached to the centromere of each chromosome. And each spindle fiber consists of 4 to 20 microtubules. Each microtubule is made of protein tubulin. That means, so this is a spindle fiber. This spindle fiber is formed by these microtubules like this. By what process? By process of a polymerization. But for this polymerization of the microtubules in spindle fibers, it requires some important things, like it requires calcium, magnesium, GTP, and calcium binding protein calmodulin. Calcium binding protein calmodulin. That means in the presence of calcium, magnesium, and energy GTP and calcium binding protein calmodulin, there will be polymerization of the spindle fibers, or there will be polymerization of the microtubules into spindle fibers. But again, one thing important, if we apply colchicine to the cell, this colchicine will inhibit the polymerization of the spindle fibers. That in that case the chromosomes cannot be separated and it will lead to polyploidy and other things. And it is very important from examination point of view. So now they will move to opposite poles, and the spindle fiber, they will form spindle fibers. Now the centriole is present only in animal cells, and now after replication they are moving to either side. That means now, since they look like stars, we can tell that they form asters. That means in case of animal cells, the mitosis is in animal cells, the mitosis is amphiastral. Amphi means both aster and star. That means there are two star-like structures, amphiastral. But in case of plants, there is no centriole. So in that case, we can tell it is anastral. That means no aster. So in case of plants, it is anastral. So this is the difference between the plant and animal cells during this prophase. So now these are the different characteristics of this prophase stage. Now towards the end of prophase, what will happen? Towards the end of a prophase, the nuclear membrane and the nucleolus will disappear. That means now the nuclear membrane will gradually disintegrate, and that will gradually disappear. The nucleolus will also disappear. And what other things? The Golgi body and endoplasmic reticulum also disappears. So when the nuclear membrane, nucleolus, and the organelles like Golgi body and endoplasmic reticulum disappear, now the prophase stage is complete and is moving to the next stage, that is the metaphase stage, that we'll discuss in the next video. But we'll discuss some questions. So one question you can write: anastral mitosis. Anastral mitosis is found in animals, plants, both, none. Then second question you can write. Second question: at what stage the nucleus, at what stage of mitosis the nucleus looks like a ball of wool. So one is prophase, the other is metaphase, third choice anaphase, and fourth choice is telophase. Next question you can write. Condensation and spiralization takes place at what stage? Again here, prophase, metaphase, anaphase, and telophase. So these are the type of questions that are usually asked in different types of examinations like NEET, NIGER, and all other examinations. I hope you will be able to answer these questions. You must have enjoyed all the details of this PROPHASE. Thank you very much. Have a good day.